Thanks for the opportunity to come. I was subbed in at a reasonably late stage because Susie Wood is the the New Zealand rep, but she had other commitments, so I've been sent along in her place. Oops, too hard. Too soft. Do this. So I'm a scientist at Cawthron. The Cawthron Institute is sort of a reasonably unique, well, not a reasonably unusual institute in New Zealand in that we're not government institute and we're not a university. We're founded by an act of parliament after Mr. Cawthron left the equivalent of about $20 million to set up this institute to do research. The institute started off working on apples and soil science, and then it's gradually morphed into doing water quality monitoring, environmental monitoring, biosecurity type questions. So my taxonomic interest, New Zealand freshwater fish, invertebrates, parasites, and basically anything we can get funding for. Because in spite of being a charity, Cawthron still expects to make a profit, or a surplus rather, and so scientists have to have their salary funded to a certain degree. And I've worked mostly in New Zealand and Antarctica. In my prior barcode involvements, I like to mention the Theraptera. They're the lice that live on birds, and I like to say the lice are the fastest running, highest flying, deepest diving, most well-travelled insect in the world, and they can't really even walk, so they're doing a good job. So according to the figures from the BOLD website, there's about 71,000 sequences on the website. It's about and that comprises about 3,800 species. And to put that into context, there's about 80,000 species in New Zealand. So we've got plenty of work to do. We're not going to run out of things to do. So the New Zealand Barcode Network, that was established by Ian Hogg in about 2007. And Ian's just recently moved from the University of Waikato to work for Polar Canada. And so we've taken on the node under iBOL 2. Barcoding in New Zealand, there's probably about 20 researchers involved, but listening to some of the talks, there's a few more people that I didn't realise were involved with barcoding through these collaborations with overseas for people. And we guessed at a number of about $100,000 that's being used for barcoding, but most of it's not dedicated. Barcoding bolted onto other projects. Future prospects, it's a bit blank, isn't it? It's a bit grim. But I think the future prospects are quite good, but it will depend on energy and taking the lead on getting new funding. So I thought I'd demonstrate some of the projects that we've got going. So the first project is a molecular tool for detecting freshwater fish. There's a project I'm involved with, and it's an environmental DNA project, and it's driven by local government water quality monitors. So they want a method other than electric fishing and netting to go out and detect what fish are swimming around in rivers and lakes. So the hope is we can take a water sample, filter the water, collect the environmental DNA and work out what fish are in there. And so once again, we have, and several people have alluded to it, but you have sort of a practical application, but you need that database to underpin the practical application. So we're going to sort of do stealth barcoding, I guess. The next example of project that our guys are doing, this is Susie Wood's project. So New Zealand's been isolated from other land masses for probably about 80 million years. And then the big factor with New Zealand is that humans have only arrived probably within the last 800 years. So there's lots of interest in what was New Zealand like before humans arrived, and how can we manipulate conditions so that we can restore some of that unique biota that's been lost after humans arrived. So this project, people are taking samples to characterise the lake communities that currently, but then also they're taking core samples, extracting DNA from those sediments to work out what was there historically, and to see if we can manipulate nutrient conditions and water flows into the lake to try and restore back to what was there in the past. Also, with this unique biota, New Zealand's got a really strong focus on biosecurity, especially marine biosecurity, and some of the regulations around international shipping are changing so that ships are required to be much cleaner than they have been in the past, which results in more juvenile growth on the hulls of ships, and it's much more difficult to identify juveniles. So this is another opportunity to do meta-barcoding and identify the communities that are coming into New Zealand on these ships. And so as part of that, Xavier has been leading a project to look at when you take a sample for meta-barcoding, what variation is there amongst all these different labs. So he's taken a standard sample, distributed around 12 different laboratories, and he's currently analysing the data to see how those differences, or how each lab, or what kind of noise each lab introduces into the analysis of the data. And then we've got another similar study where they're looking at the biota that comes in ships' ballast water. So a lot of ships come to New Zealand reasonably empty, and they ballast the ships by filling them with water. And then the ships are supposed to pump out the water 12 miles offshore, about 30 kilometres offshore, to prevent establishment of new species. So to know how effective that is, they really need to know what's in the ballast water. So we've been doing meta-barcoding of ballast water samples. And then this is a project that I'm quite excited about. So one of the issues in New Zealand is that New Zealand has the 4th biggest exclusive economic zone in the world, so the marine zone. And New Zealand also has a territorial claim on Antarctica, and it's really difficult to sample those two environments, Antarctica and our enormous marine zone. So we're looking to establish vessels of opportunity, or establish methods to sample from vessels of opportunity, so fishing boats that are going down to Antarctica to fish. But also New Zealand has a base in the Ross Sea, and most of the drinking water or the fresh water on the base comes from a reverse osmosis plant, which they take in seawater from just off the base. And this reverse osmosis plant has a pre-filter, and it collects enormous numbers of marine invertebrates. And what we're working towards is getting a series of permanent sampling points established on the Antarctic bases, because most of the other Antarctic bases use a similar system. And so you can see this little pre-filter. It's just basically a sock, and all it does is collect marine invertebrates. And so it's really easy to just empty that out and look at what taxa are in there. And Mary Sewell, who's leading this project, she's already got some preliminary data looking at morphology, but because a lot of the taxa are undescribed, it's an ideal opportunity for meta-barcoding and for just ordinary barcoding as well. And so we're hoping to get some kind of collaborative project going so we can sort of go right around the continent. So the SWOT analysis, the strengths, there's quite a strong acceptance of the need for barcode libraries in New Zealand. So recently this was just published. This is a decadal plan for taxonomy in Australia and in New Zealand. And you can read there that they're really strongly supporting DNA sequencing to help complement traditional taxonomy. And this is an ideal opportunity to use this plan to leverage funding. So that's a strength. The weakness is a lack of funding. And while I've been sitting here, I realise I kind of just underestimated the weaknesses and the threats, or that they were just lack of funding, because we're also facing issues with, as New Zealand moves from a kind of post-colonial system of government, we're getting much more into collaborative administration of resources. So the first settlers in New Zealand, or first human settlers in New Zealand, were the Maori, and they have quite strong concerns about sequencing and sending material offshore, and they're pushing towards getting some kind of system where they get to have the final say whether we can send samples or not. And it's very difficult to work with them because there's about 30 different tribes, and they all have a mandate over different territories, and they all expect to have some input into anyone who collects material from their areas. So that's a threat, but there is quite a good opportunity as well in that we have a good list of the species that are in New Zealand. So we have this New Zealand Organisms Register. It's a searchable online database, but it's currently unfunded. And I think, not to be mean to the traditional taxonomists, but morphological taxonomy on its own is not viewed particularly as a good use of funds in New Zealand. So if we can get this DNA and morphological taxonomy working together, I think that will increase the case for this funding of this register. And with that, thank you very much.